let's get right into it. If you're gearing up for the IGCSE Biology Paper 6, you know it's a different kind of beast. It's all about those practical skills. So in this explainer, we are going to break down exactly what you need to do to walk into that exam feeling confident and ready to go. And hey, if you're feeling a bit stressed, you are definitely not alone. I mean, check out this quote from Reddit. It pretty much sums up how a lot of students feel. But don't you worry. The best thing about Paper 6 is that it's not about memorizing a textbook. It's about a specific set of skills that you can absolutely learn and master. So let's get started. First up, we're going to take all that exam stress and turn it into a super focused strategy. And the way we do that is by understanding what the examiners are really looking for. So what is this paper actually testing? It's not just a memory dump of facts. See, they want to know if you can think like a scientist. Can you plan an investigation from scratch? Can you handle data, with tables and calculations? Can you draw a perfect graph? And can you spot ways to make an experiment better? It's all about the process. Okay, let's talk about the big one. That 6-mark question that asks you to plan a whole investigation. It can seem pretty intimidating, right? But what if I told you there's a simple blueprint, a kind of cheat sheet that you can use every single time? And here it is. This 7-step plan is your key. If you can talk about these 7 things in your answer, you're pretty much golden. You've got to name your variables, explain your method, include a control, say what you expect to happen, and, this is a big one, list your safety precautions. Hit these points, and you are well on your way to getting those top marks. That's the million-dollar question, isn't it? Getting your variables straight is like the foundation of your entire plan. If you mess this up, the whole thing falls apart. So let's make it crystal clear with a really common example. This makes it so easy. Think about that classic osmosis experiment with a potato. Your independent variable is the one thing you decide to change. So in this case, it's the concentration of the sugar solution. The dependent variable is what you measure to see the effect of that change. So you're going to be measuring the change in the potato's mass. And then you have the controlled variables. This is everything else you have to keep the same to make it a fair test. Things like temperature, how long you leave it, the size of the potato pieces, all that stuff. So let's put it all together and walk through one of the most popular experiments they could ask about, the osmosis practical. We're going to look at the key steps and, of course, those super important safety rules. But first, a quick refresher. What even is osmosis? In simple terms, it's just water moving. Water molecules naturally move from a place where there's a lot of them to a place where there are fewer of them across a special barrier, like a cell membrane. Getting this definition down is crucial for explaining your results later. Okay, step 1. As you can see here, you take identical pieces of something like a radish or potato and pop them into solutions with different concentrations. It is absolutely essential that the pieces are the same size and mass to begin with. That's one of your controlled variables, remember? Then you wait. After a set amount of time, let's say 30 minutes, you take them out. And look what happened. The radish in the dilute solution got heavier because water moved into its cells. But the one in the concentrated solution, it actually lost weight because water moved out. That's osmosis, right there, in action. And you absolutely cannot forget safety. These are easy marks, so don't leave them on the table. For an experiment like this, you'd talk about being careful with sharp tools like a scalpel, wearing safety goggles to protect your eyes, and mopping up any spills right away so no one slips. Simple stuff, but so important. So you've done the experiment, but you are not finished. Nope. Now you've got all this raw data, these changes in mass, and you have to process it. You got to turn those numbers into actual marks. And the way we do that is by calculating the percentage change. So why do we do this? Why not just use the change in grams? Well, because calculating a percentage standardizes everything. It lets you make a fair comparison, even if your starting potato pieces weren't exactly the same weight. And here is the formula you just have to memorize. It's final mass minus initial mass divided by the initial mass, and then you multiply all that by 100. Super simple. If you get a positive number, it gained mass. If you get a negative number, it lost mass. Easy. Okay, so you've done your calculations for every solution. You have a neat table full of data. But how do you actually show the trend? What's the best way to see the pattern and figure out what it all means? You draw a graph, of course. And listen up, because these rules are your ticket to getting every single mark. The independent variable, what you changed, the solution concentration, always goes on the x-axis at the bottom. The dependent variable, what you measured, the percentage change, goes on the y-axis. Use a sharp pencil, plot your points with neat little crosses, use as much of the graph paper as you can, and please, 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 do not forget to label your axes with units. We are on the home stretch now. Let's pull everything together, clear up a really common mistake, and then finish with some quick-fire tips to make sure you walk into that exam feeling like a pro. Okay, quick heads up on a classic trip up. People always get confused about what happens to plant cells versus animal cells. When a plant cell loses water, its membrane pulls away from the cell wall. We call that plasmolysis. But in pure water, an animal cell, which has no cell wall, will swell up and literally burst. That's called lysis. A plant cell just gets nice and firm. Knowing that difference is an easy way to pick up extra marks. And finally, a few last bits of advice. Make sure you know your food tests. Brush up on how to calculate magnification. And seriously, do a couple of past papers. You'll start to see the same types of questions pop up again and again. Read the question carefully for little details, like how to round your answers. And remember to put units on everything. But most of all, try to stay calm. You've got this. So, as we wrap up, I'll leave you with this question to think about. All these skills we've just covered, planning, controlling variables, measuring, graphing, they're not just for osmosis. Where else could you use this exact same framework? Because once you master these core skills, you haven't just learned